Thanks for inviting me to participate in this amazing symposium. These are my disclosures. I want to start by acknowledging Ben Cow, who generously shared some of his slides with a wide number of individuals that includes some data that's not publicly available, and I'm going to share some of that information with you. I also want to share with you a long list of Penn investigators that have been involved in designing and helping to implement the studies, some of which I will share with you, at least their designs. We don't have any data yet. And I want to give a shout out to a couple of amazing project managers that really make this work possible. So here's what I hope to accomplish in the next few minutes. I want to give you an idea of how I think about the clinical trials that we're conducting for COVID-19. I want to share with you some of the limitations, some of the ways that I think we should be doing it, because when I think about the trials, I always imagine the optimal universe of how we would treat people were we going to have effective agents. And that's the way I think about how to design the appropriate trials. I want to share with you the limited clinical trial experience we have to date, what we're doing at the University of Pennsylvania, and then I'll wrap things up. So first, I think it's important to understand the trials in the context of the clinical disease. So this is data from a cohort from Beijing. And the important thing to realize here is that individuals who are hospitalized generally have symptoms for about a week before they are admitted to the hospital. And thereafter, their disease progresses actually quite rapidly, so that within a few days, individuals may often be admitted to the intensive care unit. The important point here is that we are not treating people during a very important period of time that we may have the greatest opportunity to limit the progression of disease. Another way to conceptualize this is this type of pyramid. And Dr. Perlman alluded to it before, and the numbers that I'm going to share with you are a little bit different because I think when all is said and done, we are going to learn that a lot more people were infected with SARS-CoV-2 than we appreciate, and a lot of people probably have very mild or asymptomatic disease, which they never sought help for. So some of the estimates are about 30% of people have no symptoms, about 60% of people have symptoms and are cared for at home. Don't think that we don't need to treat these individuals because they will survive their illness. I've spoken to a number of people, some of whom are my friends, sick at home with this disease. They are not happy people. They wish they had an antiviral. And it is conceivable that some of the lung disease that they will get will be associated with bad outcomes in the future. So this is not necessarily a benign viral disease, and people will recover at home and everything will be fine. I think about 10% of individuals with this disease will get hospitalized. About 25% will wind up in the intensive care unit. And as more and more people are getting hospitalized, the proportion of individuals with more severe disease is increasing. And we are sending people home who probably should be hospitalized, but there is not room in the house to accommodate everybody. So it's only a relatively small percentage of all of the infected individuals that will receive any treatment whatsoever. I think about lessons that I have learned in the treatment of HIV infection and influenza and how it could be applied to what we're doing to treat COVID-19. And I think here are a couple of important things to remember. First, in the context of HIV infection, HIV replication drives all of the pathogenic events in HIV disease. I think it's a similar thing here too. I think SARS-CoV-2 is driving every single pathologic event that we see in people with COVID-19. And therefore, we need to stop virus replication. That's got to be our first therapeutic priority. In influenza, we have learned that early treatment has the greatest impact in preventing the extent of disease. And so we should be treating people long before they're hospitalized. We don't have the tools to do that now. And finally, for HIV, we've learned that treating HIV doesn't only have benefit for the individual, but it has a public health benefit. And I think if we could treat more people with COVID-19 who are not hospitalized, that would go a long way to prevent other people from becoming infected and not wind up in the hospital. So if you are looking at some of the clinical studies in people who are treated for this disease, there are two important things to think about. First, clinical endpoints. Can we prevent people from winding up in intensive care units? Can we shorten the hospitalization? Obviously, can we prevent death? But stopping the shedding of virus is also a very important goal. And if we had a therapeutic that didn't shorten the course of illness, but stop viral shedding, that would be important from a public health perspective. It would probably prevent some individuals from becoming infected. So this morning, as I was finishing up this talk, I went on the ClinicalTrials.gov website to see how many clinical studies have been registered for COVID-19. And the number is 143. So that is not a small number of trials for any disease. And we are really just in the very beginning of understanding some of the results from these studies. So I won't be able to share a lot of results with you, but I'll have some. Let me refer you to a great short summary article in Science magazine that talks about some of the antiviral strategies. You've heard a little bit about this before. There's a lot of interest in looking at antibodies or plasma to prevent fusion of the virus to the ACE2 receptor. We are interested in looking at chloroquine and hydroxychloroquine, which probably inhibits virus replication through interacting with lysosomes during the endocytosis process. Lopinavir, ritonavir, an HIV protease inhibitor, has been tested against SARS-CoV-2. Remdesivir, an RNA-dependent RNA polymerase, has been suggested by other speakers as perhaps one of the most promising agents to treat this disease. And I think that's probably accurate. So let me share with you some data. So this is one of the larger studies of lopinavir, ritonavir. It's a placebo-controlled comparison with an outcome of time to improvement. This is a study that was done in Beijing. And they studied about 90 individuals who were started on study with 12 days or less of symptoms, and another group, a little bit over 100, who started on therapy a little bit later. You can see from this time to event analysis, time to clinical improvement, that lopinavir, ritonavir did not shorten the time to clinical improvement. So one of the important clinical endpoints, clinical endpoints, was not realized in this study. And here I'm showing you data looking at the impact on virus shedding of lopinavir, ritonavir, and the control population. No difference in the reduction in viral load in nasopharyngeal swab specimens. So this drug does not shorten the time of viral shedding. I think we should not be studying this drug further. Here's some recent data from a MERS-CoV mouse model of coronavirus infection that compares remdesivir treatment, lopinavir, ritonavir, other antivirals, interferon versus placebo. And what I'm showing you on this slide is the change in weight that animals experienced after they were infected and they got a placebo or remdesivir. You can see that the animals that got remdesivir, these are the animals that are represented in green, maintained their weight, while the animals that got the placebo lost weight. And if you look at the titer of virus that could be identified in lung tissue 2 days or 6 days after the infection, you can see that early on remdesivir has an antiviral effect, but after 6 days the effect is more profound, with about a 3+ log reduction in viral titers in the lung compared to animals. So a very promising agent. There is a phase 3 study that was done in China. This study has been enrolled, and a manuscript is now under review. The trial compared Remdesivir, 10 days of intravenous dosing. Remdesivir is an intravenously delivered product versus placebo. These were hospitalized patients with less than 12 days of symptoms who met some safety criteria with respect to renal and liver function. So we will see in this study clinical events that are reported. And at Penn, we have 3 different studies of Remdesivir that are underway. An NIH-sponsored adaptive design trial, the initial phase of which compared Remdesivir versus placebo. It included 400 individuals. Bill Short and Pablo Tebas led the study here. The study will now be expanded to 700 individuals, and the next phase will probably have some immune modulating arm added to it. This has both clinical and virologic endpoints. We've got 2 Gilead-sponsored studies, one in severe disease, which initially compares 5 versus 10 days of therapy, and in moderate disease, 5 versus 10 days of therapy, and also a placebo in this arm. It's clinical endpoints that are being reported on in this study. There's a lot of interest in hydroxychloroquine and chloroquine as an antiviral agent to treat COVID-19. This is some in vitro data that establishes the proof of principle of these agents as potentially effective in this disease. And in this analysis, hydroxychloroquine outperforms chloroquine. Sarah shared some of her data with you a little bit earlier. And I want to share with you some data from a very preliminary French study that has gotten a lot of attention, I think somewhat unwarranted. This is a non-controlled trial with 3 different sites in France. One of the sites gave active agent or a placebo. The other 2 just gave placebo. And I'm showing you data on just 36 individuals. Now, just keep in mind the magnitude of the disease that exists in France and other European hospitals. There are a lot of patients. So if we are just selecting 36, and this is not a randomized sample, it is very difficult to know that the baseline characteristics of the people who get the active agent or the placebo are the same. There's been a huge amount of attention looking at this particular combination of hydroxychloroquine and azithromycin with the suggestion that this arm cleared virus from nasopharyngeal specimens more rapidly than did hydroxychloroquine or was seen in the control arm, but this is only 6 individuals. So I think it is far too early to make any definitive conclusions about that particular combination, and we need more control data to understand the effectiveness of hydroxychloroquine in this population. Ravi Amaravati is leading 3 different studies. Ben Abella from our emergency department is doing a lot of work in support of these trials. These are termed the patch studies. One study in hospitalized patients will compare higher versus lower doses of hydroxychloroquine. We will be treating some people who are at home and who are never hospitalized. I think this is really important. It would be a major step forward to show that we could decrease the duration of symptoms and maybe shorten the time people are in quarantine if we're treating people at home in this randomized study, or what we will learn from this randomized study. And then importantly, we're going to be doing one of the first preventative studies in healthcare workers comparing hydroxychloroquine versus placebo. These 3 trials are IRB approved, and they're going to start pretty soon. I suspect early next week. I'm going to switch gears a little bit. I want to talk about antibody therapy for a moment. There are some SARS-CoV-2 specific monoclonals that are being developed but I'm sharing with you some very early data in people who were treated with a humanized anti-CD147 antibody. CD147 is a multifunctional transmembrane protein. It's not clear to me precisely how this antibody inhibits entry of virus or fusion of viral and cellular membranes. But in a small number of individuals, I think it's 17 people who get the active drug and 11 who get the controls. Again, very small numbers here. We see more rapid clearance of virus in nasopharyngeal specimens to negative in the people who get the active drug, and more rapid discharge of hospitalized patients. There's a lot of interest in collecting plasma from individuals who have convalesced from their infection, and we have some really early data in 5 people, just 5 people, who received convalescent plasma from donors who were asymptomatic following their infection for 10 or more days, had a negative nasopharyngeal swab obtained, and had high ELISA titers, SARS-CoV-2, with a neutralizing antibody titer greater than 40. These were hospitalized patients, mechanically ventilated, really sick, and there's an assortment of endpoints in the study, and I will share with you 4 of them. Cycle threshold refers to the number of cycles in the PCR assay after which the test becomes positive. So then the more cycles, the less virus you're detecting, viral nucleic acid you're detecting. SOFA is a end organ clinical scale looking at organ failure, so lower on the scale is better. PaO2 to FiO2 ratio, the higher the better, and temperature, obviously, the lower the better. And for each of these outcomes, there's a suggestion of improvement over time. But remember, this is an uncontrolled study, so it's hard to make a lot of conclusions from the experience of 5 individuals. Penn is rapidly moving forward with a convalescent plasma therapy trial. We have a donor protocol, IRB, that's been approved. We have a treatment protocol in development. There's a website that I'm showing you here that is now established, and individuals can register on this website if they're interested in donating plasma. Our criteria for being a donor are a little bit different than what I showed you in that previous study. People need to be 14 days asymptomatic, no transmissible diseases. We've got a lot of interest from people who are recovering from this disease, want to develop their plasma, and we will be happy to take it. I'm going to switch gears a little bit, and I want to talk about some other adjunctive types of therapy. So the Renin-Aldosterone-Angiotensin system is really important in COVID-19 pathogenesis. As has been mentioned in earlier talks, ACE2 is the receptor for viral entry, but ACE2 levels also have an impact on the degree of inflammation in the lung, with higher levels being protective. So we don't know at this point in time whether ACE2 is going to be protective or a worsening factor with respect to the severity of disease. And Julio Chirinos and Jordana Cohn from our renal division have developed a protocol where they'll be looking at individuals with a history of hypertension, either on an ACE inhibitor or an ARB when they come into the hospital, and either continue them on therapy or stop their therapy to look at whether or not that kind of treatment decision has any impact on clinical events. We have heard this morning and this afternoon talk about the cytokine storm associated with more advanced COVID-19 disease. And from what Nuala Meyer, our ARDS guru here at Penn, tells us, that the level of IL-6 that we see in individuals with COVID-19 is not as high as what is seen in the people, in comparison to the people who have cytokine storm following the receipt of CAR T-cell therapy. So it's not clear to me whether we're just seeing cytokine dysregulation, some slightly higher IL-6 levels that we may see in individuals with sepsis or other severe illness. And there's a whole slew of immune modulating products that are being proposed for study with this disease. We have a committee that is sorting through some of these concepts and establishing priorities. And as I said, NULA is really helping us a lot in this particular regard. So I think if we're looking to understand the ideal approach for clinical trials for COVID-19, what we need is first stop virus replication. A lot of people who are really sick shed virus for a long period of time. We need to stop virus replication. Second thing is we need to understand how we should be modulating the immune response in people with severe disease and whether or not that will be any benefit at all. And I think really critically important is we need treatments for individuals who are less symptomatic at home with disease to try to decrease the spread of illness in the community.